Hello, everyone, and welcome to today's webinar titled Multiple Biomarker Analysis Made Easy with Luminex Multiplex Applications. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I'm Antonina Salcido of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Luminex, a Diasorin company. For more information about Luminex, please go to LuminexCorp.com. Now let's get started. Before we begin, I'd just like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Stephen Angeloni, Senior Field Application Scientist at Luminex Corporation, and Dr. Jacqueline Surls, Director of LuminexPLORE Labs at Luminex Corporation. For a complete biography on our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Angeloni and Dr. Surls, welcome. You may now begin your presentations.
Thank you very much for that introduction, and welcome to all you viewers from wherever you are. If you don't know about Luminex, we're going to be diving into that in more detail here. So, multiple biomarker analysis made easy with Luminex Multiplex Applications. Luminex has a long history, over 30 years of helping researchers in basic research, translational research, and therapeutics in many different fields. While the company started in Austin, Texas in 1995, it's able to grow expansively with servicing these basic research and clinical diagnostic teams or applications, and we've grown to have over 1,150 employees globally. We were a publicly traded company until 2021 when we got acquired by Diasorin, so now we're a privately held company. But in this time, we were able to grow globally and have offices on several continents around the world, with our Luminex headquarters for xMAP technology still being based in Austin, Texas, and the Italian headquarters being just outside of Milan in Italy. Now, instead of building a lot of assays, Luminex started with the method of hiring, or I should say licensing, our technology to other partners who had expertise in very many fields so that they could develop unique multiplex assays that could be run for all kinds of different applications. So we currently have over 70 plus partners and licensees that are developing assays and have kits that are ready to use off the shelf in a variety of different disease categories and applications, as you can see in the list here. Now, all of these assays can be run on any of our instruments, and these are some of the typical instruments that are used mostly for our assays. The LX200 and FM3D are instruments that have been around for a couple of decades now. And then recently, we introduced this new line of instruments called the iFlex instruments, which has everything more integrated into one unit, as opposed to the FM3D and LX200. It has computers and monitors outside that take up more desk space on your lab bench. The iFlex also comes with another enhanced feature with the dual reporter version, where you can also look at second reporters. So you can now look at a PE reporter and a purple laser blue emitting reporter. So now you can look at two analytes in the same reaction on each bead in one well. These three instruments here, the IntelliFlex, the FM3D, and the 200, they're all flow-based instruments. The LX200, however, it has only one sample loop, whereas the FM3D and both iFlex, they have two sample loops. And the advantage of having two sample loops is the run times on a full 96 well plate can be on the order of 17 to 20 minutes with the iFlex or the FM3D. It can take closer to 45 minutes to an hour on the LX200. So if you have lots of samples to process, you're going to be wanting to probably have at least an FM3D or one of the iFlex models. Now, with that partner mode of doing business, we have several partners that have a number of ready-to-use off-the-shelf kits. And most of these are proteomic-based. They're mostly sandwich capture chemistries. And in total, across all of our partners, we have over 1,000 analytes that you can pick from using our kit finder to determine if there are certain protein analytes that are in some of these multiplex kits that you might want to use for your applications. Now, one of the things that makes the platform so versatile, as I'm going to show here to go an overview over how the platform works, is the beads have these carboxyl groups on the bead surface. And what makes this versatile is that when you activate the carboxyl group with EDC, any compounds that have a free primary amine, such as a lysine on a protein molecule or an amine-modified oligonucleotide, they can be coupled to the activated carboxyl group, which allows us to develop both proteomic and genomic assays on the platform. So for proteomic applications, there's a variety of different chemistries that can be built. You can build sandwich capture assays, there's competitive assays, and over the last 2 years, the indirect serological assays, where you have an antigen coupled to a bead, and you're looking for antibodies in plasma or in serum, or if you're designing recombinant antibodies and you see how specific they are for an antigen. This type of assay has been used quite extensively the last 2 years during the pandemic. On the genomic side, when you couple oligos to the beads, capture oligos, you can do genotyping. So this is an example where we're looking at 2 alleles, and this is indicating that it's homozygous A. But you can also develop gene expression platforms, copy number variation. You can do disease diagnostics. And we also have a unique chemistry for doing microRNA detection as well. The other power that adds to the platform is the multiplexing capability. And the way we do that is we have 3 different dyes that the bead can be impregnated with. We actually have 500 different mixes of these dyes where there's different ratios of the dyes in each mix. So when a batch of beads is dyed with one of these mixes, it gives it a unique color compared to all the other dye batches that are done with a different mix of this. So essentially, with the 500 different dye combinations, we have 500 bead regions of different colors. And we usually refer to these as a bead region or bead ID. So the way multiplexing is achieved is that each bead region or bead ID is going to have a molecule that's specific for capturing a certain analyte. So when you have a number of these different bead regions mixed into one reaction, where each bead is capturing a different analyte to report to you on the level of that analyte in the sample. What this does now is it allows you to have in each well of a 96-well plate literally up to almost 500 different markers in that well. And if you have one of our FM3D or iFlex instruments, you can also do the same thing in 3 to 4 well plates. So what are the advantages of multiplexing? The first thing is you're going to use a lot less sample, you're going to use less reagents, and you're going to get more accurate data and get it faster. And so here's some real examples. This is an example of a 110-plex gene expression assay that we built for the University of Tokyo. It actually had 107 transcripts and 3 control bead probes in it. And when we ran that assay with 14 samples in triplicate, we only required 42 wells of a 96-well plate. But if you did the same chemistry with TACMAN or some real-time PCR single-plex chemistry, that would have been almost 4,500 wells, which is almost 47 96-well plates. On the proteomic side, this is an example of a real assay that we ran from one of our partners, and the client was running it as a 24-plex, but it could also be run as a 43-plex. No matter what plex you're running with Luminex, when we were running, say, this 24-plex with 27 samples in triplicate, it only required 41 wells of a 96-well plate. And as I said, that number of wells would still be the same, whether it was 24-plex, 43-plex, 10-plex, whatever it was, it'd be the same number of wells on a 96-well plate. But if you did this with a single-plex ELISA, the 24-plex alone would have been almost 2,000 reactions, which is a little more than 20 wells. And if you bump that up to 43 markers, you would then be in the realm of almost 3,500 reactions and a little over 36 96-well plates. So with this platform, whether you're doing a proteomic or genomic assay, you can literally go from something like this, where you're literally running dozens of plates, to now having everything here in one plate. So you can see that with all your analytes in one reaction, you're going to use a tremendous amount of less sample. The amount of reagents you're going to do is going to be significantly less. And because all your sample is not spread out amongst multiple wells, where you could have incorrect loading, you won't have the problem where if a sample is not getting loaded correctly, or if the reaction reagents, detection reagents are not loaded correctly, any shift in signal will not happen, what could happen with individual wells. But in the multiplex assay now, you have all the analytes in one reaction. So if you did, do a, and I'm in a hotel room and somebody's coming in, hold on. Excuse me, doing a call. Oh, yes, come back. Yep, sorry about that, everybody. Should have put the no do not disturb sign out there. So if you have all these in one reaction, then you're going to use a lot less sample. And if anything is off, if you're doing in triplicate, you'll know. Sometimes, however, you can actually have certain control beads in the well. So if you don't want to run duplicates or triplicates, the control beads in the well can monitor the effectiveness of that reaction. And you'll know right away, even with using just one reaction, if you need to repeat that sample or not. So why test different types of biomarkers using multiplexing? So one of the reasons why you should use the platform is that biomarkers for testing the stability and characteristics of production cell lines are required because you have to validate the purity and the authenticity of the cell lines. It's very important when you're using cells as bioreactors to know that your cells are going to be pure and they're going to be the correct cell type when using them to generate biologicals. Now, the ATCC put out a publication on this because they biobank a lot of cell lines, and they've realized over the years that some people have cell lines that aren't really what they think they are. So you need a method to authenticate that cell line. And if you have cell line stocks and you pass them over time, you want to monitor them for being the same, all having all the same characteristics with each passage as you bring them into the production line. The other thing is that you need to check for contamination. This is contamination with any microorganisms, bacteria, yeast, fungi, viruses. You want to make sure your cell line is not does not have any contaminants and is pure and is the right cell line. The other reason is that the FDA has certain guidelines for biological products used for clinical applications. And in these products, you want to make sure that during the process, again, everything is pure and not contaminated and is consistently making the same type of product with the same level of purity each time. So while this guidance here focuses on viruses, it is also applicable to other types of biological production, such as any proteins or biologicals that are made with other types of host cell proteins or host cell expression systems. Whenever possible, the contaminants introduced by the recovery or purification process should be below certain detectable levels with the highly sensitive analytical method. So again, this includes, any viruses, any metabolic changes that might be happening in your cell line that could impact the production of your final biological product. The best way to monitor these is with multiple types of markers instead of maybe just one or two markers. So in order to do this, you can do this with genomic applications. So as that one paper described with regards to any type of non-cell specific contaminants, such as bacteria, yeast, fungi, viruses, whatever, you can develop a variety of genomic applications that can target that, as this paper exemplifies. And then as far as determining, authentication of your cell line, to make sure if it's a human cell line, it's the correct human cell line, and it's not contaminated with other cell lines, we can develop genomic assays to do that type of analysis as well. On the proteomic side, again, with our partners who have a variety of kits you can look up with our kit finder, they have, like I said, almost over 1,000 protein targets. And one of our partners, Thermo, even has a genomic assay that can be used for certain applications as well. And each of these partners, they have sites where you can go to the partner site and look up what the kind of kits they have. So, for example, the Cardiplex assay from Thermo Fisher has over 600 cytokines, chemokines, and growth factors and other target proteins for mouse, human, rat, and some non-human primate, as well as other species, for looking at the production of these proteins in multiplex format from a variety of species. And in this case, the plex goes from anywhere from 2 up to 80 plex. So Bio-Rad also has a number of kits, and they have up to almost 500 targets for mouse, rat, human, and a number of other species as well. MultiPLORE also has a large selection for a variety of species, and they go up to almost 600 markers as well, with all their different applications, for some of which are targeted for specific diseases, as well as being customizable to a variety of other needs. And then Bio-Techne is another one of our partners that has on the order of 450 analytes and growing that can be used to detect these proteins in a variety of different species. So, resources for developing multiplex assays. So one of the first resources you can look at, especially if you're a do-it-yourselfer, is the xMAP cookbook. The xMAP cookbook has a variety of protocols and recipes for building a variety of genomic and proteomic applications. Now, while it seems like these are easy to follow protocols, one thing I have to warn you is that they are a little more complicated than you think. So if you're interested in building an assay, we would recommend, if you want to do this on site with your own reagents, that you contact us to request on-site training, where we can come into your lab and spend up to 4 days or almost a week and help you build an assay and have a prototype assay working in your hands within about 4 days. Another option is that we have a number of online resources and courses you can take down in Austin, Texas, to train you on how to use the instruments as well as to build certain types of assays. If you don't have the manpower or the time to do this yourself, we also have the xMAP Lab, which is a service down in Austin, Texas, that Jackie heads, and I'll turn the presentation over to her so she can tell you about the options with the xMAP Lab opportunities there.
Thank you, Steve. All right. So I'm going to talk to you a little bit about LuminexPLORE Lab. What is LuminexPLORE Lab if you haven't heard about it already? So our lab provides research services with Luminex xMAP technology for life sciences. Our team is composed of highly skilled and diverse application scientists across multiple disciplines. Our mission is to enable your success with our technology and to help you get your information, your assays or products, whether they're a vaccine, a therapeutic, a diagnostic, faster to market. And as Steve mentioned, our technology can cover a multitude of applications, and looking at the stability of your production cell line, looking at the purity of your products, quantitative measurement of HCPs, and looking at multiplex genomic assays to test for looking at the genetic specificities and contamination. So, how can xPLORE Lab help with our team? We can do a quick statement of work proposal, helping you with our assay services. We can build an assay for you for HCP profiling in our own lab or at your lab using our FAS scientific team. We can work with your design. Let's say you built your own assay, and we can work with that design and help you optimize it. Anything that we build in our lab, we can do a simple design transfer back to you. So, we would hand you back all the protocols, everything that we did to build it, and give it all back to you or any CRO of your choice. And that will help enable you to reduce your project timelines. We have an extensive array of resources. We have a team of experts with diverse backgrounds that I mentioned earlier that can help you provide comprehensive support. So we have lab manager, project managers, scientific team, a dedicated lead scientist that will work with a team of scientists to help you meet your goal. Now, as Steve mentioned, there's a number of partner kits available that may already have a target of interest for an HCP. And, we've kind of looked at some of those already. If you look in the literature, some of those are already available that you may not be aware of. Kind of when I've looked at the literature, ELISA is a benchmark for HCP profiling. And if you look at some of our partner kits, a lot of those are already readily available on our Luminex partner kits from the Biorad, the MilliporeLab, Thermo Fisher, and some of the others. But what happens as you get further in the development or, some of the process specific needs, if you can't find a kit that's already available, you build it. As Steve alluded to earlier with the cookbook, for those that you like to do it yourself, analogous to, playing with Legos and putting those building blocks together, it's a lot like, making a little Lego set. You figure out what you want to build and you find out the Lego pieces, what you're going to need, and you assemble it. And hopefully at the end of the day, you have a nice assay. So far what I've seen is it's a typical sandwich capture assay for the HCP profiling. And very similar to an ELISA, where you have your antibody matched pair and you have your sandwich capture assay, which recognizes two different epitopes for your target of interest. And instead of your primary antibody being coded onto a plate, you have your primary antibody coded onto a magplex microsphere, or the bead. And that bead has a specific bead ID. And so you'll be able to detect it for that specific target of interest. And your detection antibody, or your secondary antibody, is going to be conjugated to a fluorophore. So, when it gets excited, it'll have a specific emission for the green laser. So, this is a very similar to an ELISA, but the advantage to the bead technology, to our xMAP bead technology, is that, it has a very good tolerance to any matrix effects. So typically what you will see with our technology is, reduced backgrounds. So it's very cleaner backgrounds. Oftentimes we'll see better sensitivity. So if you're looking to have better sensitivity, this is what you'll often see with our xMAP technology. And so one of the things that, we hope to better understand if this will suit your needs, if you're wanting to, profile more HCPs, and perhaps this is of interest, perhaps during the different stages during your production cycle. And very similar to an ELISA workflow, You prepare your dilution series of antigens, you add your bead mix to the plate, incubation step, wash, add your detection antibodies, wash. In this case, the detection antibody isn't biotin conjugated. It has a fluorophore attached, so you're not going to add SAPE, but you'll wash, and then you're going to read the plate. And so, typical plate layout, quantitative measure you can do, you're going to have a standard curve, can have controls, and then you're going to run your samples. And as Steve mentioned, we have a multitude of instruments. Here I have an example of the IntelliFlex, our newest instrument. And I think what Steve may have mentioned is that we can do automation. So because we have magnetic beads, you can do high throughput. So if that's something that is of interest to you, you can do a fully automated system with this. So, you can run this assay in a 96 well plate format, or you can run it in a 384 well format, and you can run it, in concert with a TCAN or Beckman automation, if you want a fully automated system. And they also work with the plate washers, like the biotech plate washers, the TCAN HydroFlex. So, you can definitely work with all of those as well. So, general assay development workflow. So if you want to be the do-it-yourselfer, I'm an assay development scientist. My team are all assay development scientists. This is what we are passionate about. This is what we love to do. This is what we love helping people do. So, obviously identify your target or targets of interest, figure out your assay design, identify your reagents, and then optimize your assay. I think definitely the critical steps are reagent identification, qualifying your antibodies, validating your antibodies, and then optimization, right? Making sure that everything is optimized correctly so that your assay doesn't fall apart, right? And then validation. And if you're not a do-it-yourselfer, for whatever the reason may be, it could be a multitude of reasons. For, a lot of people are more, perfectly capable of being a do-it-yourselfer, but for whatever reason, it could be bandwidth, it could be time, it could be resources. Our lab, can definitely provide those services for you. And the way that typically works is, we have several phases, and everything is phase gated. So we have a project scope where we, definitely have a good understanding of your project. What are your needs? What are the design inputs? What are the specifications? Once we have a good understanding, and that's kind of where we do the work through the design, optimization, all of that, spec out the reagents and all of this. Then we'll do a design review. We get to phase 3, we'll do a fit for purpose. Then we'll have a stage gate and do another design review. Make sure all the design outputs met the design input and do a design verification. Then we'll enter validation and enter another gate review, and then make sure that the assay meets your specifications. And this is where we lock in the final assay configuration, where no more changes are made. If the assay met specifications, everything is completely locked in. And then we can either transfer everything to you, a CRO, or we can custom couple the beads for you. Or you can build them yourself, however you prefer. And in the statement of work proposal, everything is clearly defined by milestones and subtasks. So there's no ambiguity as to what is done, and everything as we go through each milestone and each gate review, there's go or no-go actions. I like to think of it that everything is very, risk-free on your end, because if it were me, I would want it to be very risk-free for me. So, the customer doesn't pay anything until a milestone is complete, and we don't move forward until you give us a green light. So in that way, if at any point you wanted to stop, during one of the milestones and say, okay, we want to stop, we want to take it from here, or we want to pause because we don't think, this is not headed where we want it to be headed, we stop everything. But typically, we rarely see that happen. All right. And so if you want to learn more about LuminexPLORE Lab, I have the site here. And if you want to contact us and ask any additional questions, I have our email here. And the fastest way is probably to reach out to your local sales representative and field application scientist. And I thank you guys so much for attending. And I really hope that you have some questions, because I am very interested in learning more. So we will take questions.
Thank you so much, both Dr. Angeloni and Dr. Surls, for your informative presentation. We'll now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Now let's get started. Our first question here is, are there off-the-shelf assays for biomarker analysis?
Well, as Jackie and I said, there's lots of them, actually. Most of them are proteomic. There are, like I said, one manufacturer, Thermo, does make some genomic assays. And they actually have quite an extensive library of genomic targets. And their chemistry can be used for looking at transcripts, so they can look at gene expression levels. And they can also look for specific, say, genomic or DNA-based sequences. So for some of those topics we talked about with regards to looking at, authenticating, is your cell line you're using, is it the right cell line? It can do that kind of stuff. In addition to, if you want to see a certain gene expression profile in the cell lines you're doing to generate certain targets, it can give you that information as well. And when you're expressing a target or something in a cell line, it can also look at the level of those transcripts that are making your protein of interest as well. And that's one of the advantages of being able to use, the platform for both proteomic and genomic. So there are off the kit shelves already and options.
Thank you for that. How does running multiplex assays have sample and time?
Yeah, actually, we have an attendee there who is a little louder than other people. What was that question again? Could you repeat the question again there?
Yes, my apologies. Dog barking in the background. The question is, how does running multiplex assays have sample and time?
Okay, yeah. Yeah, well, I think your dog is not as bad as the maid that was trying to come in and make my bed here. But anyway. Yeah, so as I mentioned, when you're running a multiplex assay compared to a singleplex, you're going to need a lot more sample, and essentially, for each analyte you're doing in singleplex, you're going to need a certain volume of sample for each of those reactions for each analyte. But now with all those analytes in one well, you only need one aliquot of that analyte. So let's say if you have a 100-plex assay, and you need 5 microliters of, analyte or sample, you're going to need like 500 microliters of sample to get all those data points. But that 5 microliters just by itself with Luminex can give you that same answer in one well of a 96-well plate. So that's one way it'll save you sample. And at the same time, it's the same with the reagents you have to use. You're going to use just that one well's worth of reagents instead of 100 different wells worth of reagents. And then since you're doing all that stuff with fewer wells, that data is going to be coming to you much faster than having to read multiple wells and having to analyze all those wells, put all the data together, because now you only read one well to get that data. So those are some of the ways that the multiplexing will save you time, give you data quicker, and because you're not, risking the possibility of misloading some sample or reagent in multiple wells, you'll minimize that problem as well.
Great, thank you. Another question we have here is, what type of genomic applications can be performed on Luminex?
Oh, well, that's, again, almost anything, actually. Definitely gene expression, SNP typing, copy number variation, and then we have a unique simple chemistry for doing microRNA analysis as well for people who might need that type of analysis.
Thank you. And the next question here is, what is the maximum number of markers that can be analyzed in one reaction?
Want to take that one, Jackie?
Yeah, 500 on the FM3D and iFlex. On the iFlex dual reporter, it's 500 on one channel on the green laser, and then if you think about it on the violet, another 500, so you could say it's double the data. On the LX200, it's 100, and then on the MagPix, it's 50.
Great, thank you. And thank you so much, both Dr. Angeloni and Dr. Surls. Do you have any final comments for our audience?
All I can say is, I know when I first was introduced to Luminex, I was doing single-plex ELISAs. And I was surprised at how quickly I could get a lot more data quicker. And the same thing with a genomic assay. When I first got out of academia, I was doing a lot of PCR reactions to study a lot of targets, for the studies I was doing in academia. I worked for a company that had a 100-plex gene expression assay on Luminex. And when I found I could run reaction and get 100 markers out just in, a couple of hours, I was flabbergasted. And even though they sometimes have more markers in the assay than you might want, that actually gives you more information and can help you discover new things that you didn't know before by having certain markers in there that you weren't normally looking at, and you see changes which could be important for the type of work you're doing.
Yeah, the other thing that I would, I definitely agree with Steve, and the thing that I would also like to add is that, as I mentioned earlier, is that, the matrix tolerances that I see with the xMAP assay is tremendous. And then those that, have transitioned from ELISA to bead-based xMAP technology, see a very good correlation from ELISA to xMAP, So I definitely, the workflow is synonymous, and, it's a very good correlation. And I also think that because of the ability to multiplex at a higher throughput, and also the ability to use the automation, and it just has it's a seamless transition. Should anybody be interested in making that path, we can definitely help move them to that transition and take that path forward. I think that those that have made that step are pleasantly surprised and glad they did.
Yeah, I can add to that as well, because especially for a lot of the proteomic assays, where you have a cell lysate or something like that for sandwich capture, or if you're doing a serological assay, like we've been doing a lot of these, putting various antigens on a bead. You want to see if there's an antibody titer to a certain pathogen. COVID, a lot of that lately, of course. Now we have people going into influenza and other viruses. But the thing is, you can actually minimize a lot of the matrix effects, because sometimes you have to use your sample at such a high dilution, but you still get your data, and you eliminate some of those matrix effects that you might get with other assays where you have to use your sample more concentrated. So it pays off in that respect too. And literally, when you build your own assay, or we build an assay for you, the beads come as like 12.5 million beads per ml. And if you were to couple a whole 1 ml of those beads, 12.5 million, and use them at the recommended amounts we recommend per region in a well, you literally get enough beads for like 5,000 reactions. So that makes it very economical as well.
Yeah. And one of the things is I would love to go in a deeper technical discussion for another talk, or have people reach out to us, the FAS or LuminexPLORE, to have a deeper technical discussion as to the different manipulations that we can do from an assay development perspective as to why we can help get it to a more sensitive level on a bead-based assay.
Yeah, as I mentioned during my portion, while we have that cookbook, the recipe's as simple as they look. There are a lot of things that we can do to enhance the way those assays work. So while we encourage you to look at the cookbook and maybe get an idea on what you might use, it's definitely best to reach out to us so we can look at your project in more detail and give you more specifics on how to make your assay, as robust as possible.
Thank you again, Dr. Angeloni and Dr. Surls, for your time today and your important research. Before we go, I'd just like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Luminex, a DSOring company, for sponsoring today's webinar. Any questions we didn't have time for or those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you, everybody. Have a good day.